Okay, in this basic bioinformatics session, I'm going to take you through some basic tools that we can use to analyse DNA sequences. And in this particular scenario, you've just assessed the BRAF mutation, so the BRAF V600E mutation. Now, what I want you to do is verify that these primers that I've designed would successfully amplify the same region of DNA that you've just analysed. So what you've just done is analyse the commercial TACMAN single nucleotide polymorphism assay, and that's one way of assessing whether a mutation is there or not. An alternate way of doing that would be to amplify the region of DNA that contains the mutation site and perform direct sequencing upon that DNA. And here are some primers here, forward primer and reverse primer used for PCR, and what we're going to do is just see if these primers that have been designed for sequencing this region of DNA, we're going to go through various steps just to verify that these primers would actually work before we actually go and actually try them out in the lab. And this is something that I always do when I design primers myself for my own TACMAN assays. I will go through very similar steps to what I'm showing you here, and it will teach you how to extract DNA sequences and also coding sequences. So what I've done, I've designed a forward primer and a reverse primer, and these are supposed to bind somewhere within the BRAF gene and hopefully amplify the right bit of DNA. So what we're going to do is just check that they bind in the right place and don't bind somewhere else. So the first thing I'm going to do is open up BLAST. And BLAST is a tool that we use for searching for nucleotide sequences, so we can input this sequence and it will tell us where that sequence exists on the human genome and also on any other genomes that we want to test. So I'm going to do that now. So I'll just open up that hyperlink and that should open BLAST. Okay, so you can see the web address there. This is what that link opens. Now on BLAST we scroll down to nucleotide BLAST. We can search for proteins as well if we really want to. And then this is the view that you'll be faced with, and this is where you can type in your primer sequence. So it's much easier just to click back to the document that you're working on, copy, Control C and Control V, scroll down and click on BLAST. And we'll leave that, and it will take a minute or two to search all known nucleotide sequences. Okay, so while it's doing that, it's probably a good idea to open up another BLAST window. So I'll just open up another BLAST window there. So the other window is opened, and I'm just going to now copy the reverse primer in there. And what we hope is going to happen is that both the forward and the reverse primer will bind somewhere near each other, a few hundred base pairs apart, because that's where I've designed the primers, and they should bind to the same piece of DNA. And if we know where the forward primer binds and where the reverse primer binds, then that allows us to calculate how much DNA there is in between those two primers, but it will also give us the total size of our PCR product. So BLAST is currently thinking about it. It might take a minute or two. Okay, and it's done. So both forward, that was the forward primer that I put in, and this is the reverse primer. So what I'm hoping to see is when I scroll down, at the top of this list there'll be some hits. 100% hit here. So that's the query that I put in, which was a forward primer sequence, and it says it's recognizing the Homo sapiens serine threonine kinase BRAF partial coding sequence. We've got 100% hit to that, and we've got 100% hit to various other sequences which refer to BRAF. So that's really quite reassuring, although it also matches chimpanzee, and it matches some other random stuff down here. But these are the top hits for this particular forward primer. So what we can do is now look at the reverse primer on the other tab. and hopefully we're going to see some of the same sequences. There we are, there's the BRAF partial coding sequence again, another sequence there. So what we want to do is identify a sequence that is both recognized by the forward primer and the reverse primer. Now the two that I'm interested in, the main one I'm interested in is this one here. And this is certainly, I mean, up here these are saying partial gene sequences. I'm just going to go for this one. It's basically, I know it's present on both, so it's this NG00787, and if we go back to the other side, NG00787, and this appears on both of the lists, and this is actually a large genomic fragment of chromosome 7, which just happens to be where the BRAF gene is. So both our forward and reverse primers match to, and I'll just move the window a little bit so you can see. So they both match the same sequence. So what we're going to do is open up that sequence and see what it is. It's 200,000 bases in length, but that doesn't matter. We're going to open that up and see where the primers bind within it. So all you have to do is click on that and it will open up this, and it's a file. It's sort of a reference sequence. It's been given a code. It just happens to contain the BRAF gene, and we know the forward and the reverse primers are in there somewhere, and if we scroll all the way down we get this ridiculously long sequence. Now your job is to locate where the primer is within that sequence. Now that's not particularly easy, but there are some simple tricks to do that. Now firstly, click on this button here, faster, and that will just put the DNA sequence into a format like this without all the gaps and without all the numbers. Okay, so you've now just got this long run of nucleotides. All we need to do now is find where our forward and reverse primers are within that sequence. Now there are specialist software to do this, but I tend to do a lot of this stuff working from home, and I just want to have access to NCBI and the basic tools. So I'm going to show you the very basic way of doing this. All we need is the primer sequence. So here's our primer sequence, and what we can do is just copy that, have a look for it in here by just doing Control F. So Control F is to find, and then Control V to paste. And what it's saying is that sequence doesn't exist within here. Now there's two possibilities. One, it could be that we might need to reverse antisense the primer, or two, it could be that the primer is sort of there at the end of a line and it's just not recognizing. So a quick simple thing to do is just start deleting a few bases, and what we find is the primer sequence that we're after actually just straddled one line to another. So quite often if you just type in the first half of a primer, if that doesn't match, type in the second half of the primer, and then you sometimes pick it up. So what we've just managed to find there is that one of the primers binds here, and the sequence that I have just deleted, a little bit of sequence I've just deleted, I've just taken that little end bit off there. I can easily, which is that little bit there, and the last base was that A. So we know that our forward primer binds here. Now all I'm going to do now is do a little quick trick to make your life easy. I'm going to copy this sequence out, and I'm going to go about 5 lines above, 5 lines below, because I know that I've designed this PCR product to be about 200 bases in length, thereabouts. I can't remember exactly how many bases, about 200. So I'm just copying out that entire sequence, and I'm going to put that, just for simplicity, into Word. Okay, so our sequence, that's our sequence that will contain the primer, the forward primer, and somewhere within there we'll also be able to find the reverse primer. Now because we've just copied it across, we've lost that location, so I'm just going to copy that again, copy the forward primer, go back to this version here, just for convenience I'll dump it there, and then I want to I'll just highlight the front half of that, copy that, press Ctrl F. That opens up the search function and it will identify the two places where that sequence exists. There's the first one, and the second one is actually where I've typed the sequence. So our forward primer goes TCA, ATA, ATG, and so on, right up until that A on the next line. So what we can now do is just highlight that region from here Okay, I'll just do that again. Sort of from that base there all the way along to here, we know that that is our forward primer, so I'll just highlight that in yellow. What we now need to do is locate the reverse primer. And the reverse primer, we can just copy that. Let's just dump it in here. So this is our reverse primer. I'll just stick a R there just so we know which is which, and that's a forward. Okay, so we need to find this sequence. Now the problem here is this sequence is going to be the reverse antisense. So if we want to locate this sequence in here, we don't type out this sequence into the search function. We have to type it backwards and starting from the end. Okay, so I will just demonstrate that. So this is a search function which I've just moved the window very slightly, so I can type this sequence backwards and anti-sensing it. So where it says GGT, I'm reading backwards, I'll go CCA and then another C, then a T, G, okay, so this is starting to match part of the sequence. Okay, so and as we're typing in the sequence, you'll notice it's going in this, it's highlighting bases in this direction. So basically the whole primer sequence is from about here along here. We've got, I'm reading the sequence backwards and anti-sensing it in my head, and there's a run of 5 A's there, so there should be a run of 5 T's, which you can see there. Then it should go GGCC, which I can see ends at that point there. So what I can now hopefully try to do is highlight that region. If I can get my little highlight tool to behave. There we go. So I've managed to highlight that region, and that is my reverse primer. So I'm now going to make that a different colour, red. Okay, so what we've now identified is this is the forward primer, and that includes the last A there as well. That is the reverse primer. So the distance from here, from that base to that base, is the number of base pairs in the PCR product if I were to use these primers. All we need to do now is effectively cut those out. I'll just move the window so you can see what I'm actually doing. So basically all the sequence from here to here is effectively my PCR product. Okay, I can just manually mess around with the sequence, tidy it up a little bit. Okay, and if I highlight this entire region and check the word count, number of characters on there, then you can work out you can use the word count function to check the number of base pairs. That tells you how long the PCR product is. And I've just highlighted that entire region of DNA, 223 characters. That tells me it's 223 bases. So it's just a little quick check that I do to find out what size PCR product I'll be expecting from these primers. Now this is all very well, but this is the DNA sequence. Now the DNA sequence might be introns, it could be exons, and we don't actually know that for sure. So this is the end of part 1. In part 2, what I'm going to do is take you through where we can verify that this is indeed the DNA sequence that represents the bit of the coding region that includes codon 600. So the next set of the instructions are going to be, how can we prove that somewhere within this sequence is codon 600, and therefore these primers will be perfectly suitable for sequencing to look for that V600E mutation. So that's what we're going to do next. So the next part, let's locate codon 600 within your PCR product just to verify that that correct piece of DNA is there. So all we need to do here is go to ncbi.nlm.nih.gov and search for BRAF under the gene select function. So I'll show you that in a moment. Okay, so this is your standard view at the NCBI portal, where you'll also find PubMed. So if we just scroll down here, there is PubMed's down there. Up here we can have a look at gene, and we can search for the gene that we're interested in, which is BRAF. And it gives us various different options, but the one we want is not the African clawed frog, we want Homo sapiens. So click on that link there. Okay, and this is the view that you will get. Okay, so what we now want to look for is to locate where the BRAF reference mRNA sequence is kept. So we're going to look out for something that's got an NM number. So this is just a code for a particular DNA or RNA sequence. This is an mRNA sequence, and each gene has got its own number, and there are various updates of that. So we're on 4333.6, it was on 0.4 last time I looked, so these constantly get updated, but the NM numbers are reference sequences, and there are often lots of different ones for individual genes, especially if there are splice variants. So here we are back at the page where we've searched for just BRAF under the gene heading, and we can scroll down and we're looking for somewhere that mentions reference sequence details or words to that effect. And here you are, there's a link there that says go to reference sequence details. So these are all of the agreed sequences of BRAF. And then if we can scroll down, now the general rule is with reference sequences, like with an NM number, is the one with the lowest number tends to be the originally identified gene, and then there are much higher numbers which tend to be more recently identified splice variants which include or exclude one or more exons. So if you always go for the NM number with the lowest number, and that's also called isoform 1. That was the first one that was identified. All of these others are sequences of different isoforms. So we're interested in this one, which is NM004333.6, and you can click on that, and that will take you to the actual reference sequence, the main agreed transcript of the BRAF gene. And that sequence file is here. It gives you the protein sequence there, and it gives you the DNA that codes for that particular protein, that codes for the mRNA that then produces that protein. So we've got our correct file. All we need to do now is locate which bit of this DNA sequence refers to the bit of DNA that codes for the region around amino acid 600, because that's the one that we really want to be as part of our PCR product. Now there are a couple of ways of doing this. The easiest one is to just take the reference sequence number here, copy it, so Control-C for copy, and we're going to import that into a software called ORF Finder. Now ORF Finder is also on NCBI, and it's basically an open reading frame finder. It looks very much like BLAST, and all you do is you copy in your accession number, that NM number for the reference sequence, and press return. What you can also do is copy in the nucleotide sequence in FASTA format. So FASTA format, you click there, and it will get rid of all of this stuff, all of this information. It'll give you the entire DNA sequence, and you could just copy and paste that into that box. So just copy that in and put that instead of the accession number. Doesn't really matter what you do, you'll get exactly the same result. And just click submit. And what this will do is scan that DNA sequence and look at all the possible open reading frames, which are basically regions of DNA which, if you copied them to mRNA and then try to translate them, you will get the longest runs of amino acids. So what it's done is identified a whole load of open reading frames out of this sequence. So some of these open reading frames are very short, encoding for a few dozen amino acids. Some of them are incredibly short. However, the longest one that jumps out is this one, which is 766 amino acids. We know that the BRAF gene is at least 600 amino acids because it's amino acid 600 that is changed when we have an oncogenic mutation to BRAF. So that's the one that I'm interested in, and that is open reading frame number 6, the longest one, which is by default showing in this window. That's showing the amino acid sequence that it is coming from this DNA that we've put in. Now we can display the open reading frame of coding sequence translation. If you click on that, and what it will do is take that open reading frame, starts with an ATG, start site, and methionine, and it will read that DNA and put in what amino acid that DNA codes for. Now the mutation is affecting codon 600. Now if that's base number 1 of the mRNA, or nucleotide number 1 of the mRNA, we know where amino acid 600 will be. It will correspond to approximately base or nucleotide 1800. So we can scroll down here all the way down this sequence until we get to about 1800, and there we go. That says 1801. That's a lysine, but the previous codon is a GTG, coding for a valine. We know that codon 600 is a valine. There is a GTG. That's very, very reassuring. This is codon 600. So this is effectively telling us the DNA sequence of that codes for, when copied, forms the mRNA, which when translated gives us the amino acids around amino acid 600. Now, just as a final check, I would be now wanting to see some of this DNA sequence in the middle of our PCR product. So what I'm now going to do is just do a little check to make sure that this sequence is actually within my PCR product. Okay, all I've just done is taken a little screen grab of that open reading frame that we were just looking at. So if we just go back, I've just taken a little snapshot of that little view there, just so I've got the sequence in my Word file. There we go. So this is the region here that we're interested in. So this line here that contains this GTG valine. So I'm just going to check that this sequence here is present somewhere within here. Okay, so I've just used the search function for this little sequence GCT ACA GTG, and that GTG is the valine coded codon, and that comes from this little region here. So it goes where my arrows GCT ACA GTG, and if you read this sequence backwards, it perfectly matches this sequence here. So that run of 4 T's is that run of 4 T's. So we know that these primers will amplify this region of the BRAF gene, and within that there is DNA that codes for codon 600 right in the middle of that PCR product. So if we amplified this DNA, if we used it for amplifying DNA from melanoma samples, we could then sequence this DNA and we would have our mutation site right there, and one of those 3 nucleotides are changed from a valine to a glutamic acid in the V600E mutation. And then back to the instructions document. We've done all of this. All I want to do now is confirm that we are definitely in the right place, and I want to see the mutation to see what to expect if I was to sequence this PCR product. So I just found this particular paper. It's looking at thyroid carcinoma, which has this same mutation within it, and it had a very handy sequencing trace. And what you can see is there is the GCT, ACA, GTG, that's the wild type sequence, and when that is mutated, that GTG, we get, instead of having a T there, that T has been converted to an A on one of the alleles there. So you can see there's the red and the green trace. This is using big dye terminator sequencing. That might mean something to you. So that codon has been converted from a GTG to a GAG. And if we consult any random codon table that we can quickly identify by Googling DNA codon table, you can see here is a GTG codon coding for valine, and when that T is converted to an A, that's a GAG for glutamic acid. So we're absolutely sure that we are dealing with the right bit of DNA, and armed with this bit of information, I would then be very confident in ordering those primers and knowing that they would amplify the right bit of DNA, and then if I sequence that PCR product, I would have the V600E mutation site right in the middle of that PCR product. Okay, I hope that all makes sense. The beauty of you doing it this way is, as I mentioned, there are some specialist software for doing genetic analysis. When I'm designing primers, I tend to be doing it without access to any specialist software. I like to be able to just access NCBI, gene, mess around the sequences and just use it on any free open access software. So that's why I show you how to do this, because I know you'll be able to do it from home. You won't need any specialist DNA analysis software, and you can just access everything freely. All of the resources I've just been on are completely freely accessible from any computer anywhere. Okay, so that's all from me. So you've now seen SNP analysis in the lab, and you've seen how we could amplify that same bit of DNA by standard PCR, and that PCR product would then be perfectly suited for direct DNA sequencing. Both methods are perfectly good for detecting point mutations.